Good morning. Let's discuss on some specialized and biologically important products derived from tyrosine. So, tyrosine serve as precursor for following several biologically important compounds like catecholamines, dopamine, norepinephrine, and epinephrine. These are three catecholamines, then melamine pigment, and then thyroxine hormone. Look on biosynthesis of catecholamines. The catecholamines, dopamine, norepinephrine, and epinephrine are synthesized from the dietary tyrosine in selected central and peripheral neurons and in the adrenal medulla by sequential action of enzymes in the synthetic pathway which was being first postulated by Blasco in 1939. So Tyrosine, the synthesis starts from the tyrosine. Tyrosine first converted up into the hydroxylation reaction via the enzyme tyrosine hydroxylase in the molecular presence of NADPH and tetrahydrobiopterin. And oxygen, supply of oxygen is also required here. So hydroxylation at ortho to the hydroxy group of tyrosine happens here. So this is the DOPA. Dihydroxyphenylalanine is being formed. Further, there is a decarboxylation reaction under the influence of decarboxylase, which is PLP dependent, pyridoxal phosphate dependent. Carbon dioxide is removed in this course of reaction and dopamine is being formed. And then dopamine further gets hydroxylates at this particular carbon, which is directly attached to the ring, and this is leading to the formation of norepinephrine. So, and this reaction is dependent on copper, presence of cupric ions, and vitamin C. Dependent reaction is this. Then norepinephrine further undergoes a methylation reaction at the amine groups. This amine gets methylated here, and the enzyme is N-methyl transferase (NMT), and this the SAM S-adenosyl methionine act as a donor of methyl group, which further converts up into the adenosyl homocysteine. Then this is epinephrine. Further, epinephrine converts up into the metanephrine. Metanephrine is means the methylation is happening at this hydroxy group. Further, SAM acts as a donor of methyl group. The enzyme is catechol O-methyl transferase. So further metanephrine to vinyl mandelic acid, very important metabolite of tyrosine metabolism. So this is the vinyl mandelic acid. Metanephrine undergoes the oxidation reaction via the MAO or the monoamino oxidase enzyme, very important enzyme, which leads to the breakdown of this amine entity here at this just adjacent to the carbon, and it turns into the CHOH. So this is vinyl mandelic acid. Why we are calling these catecholamines? Because all these are, while it is dopa, dopamine, norepinephrine, epinephrine, all these have this hydroxyl. Hydroxy phenyl ring means the two hydroxy groups are at ortho position. So this ring is known as catechol. So all derived from catechol ring of the tyrosine. The catecholamines, norepinephrine and epinephrine, they regulate the carbohydrate and lipid metabolism. Epinephrine increases the glycogenolysis and stimulates the lipolysis, increases the blood pressure, increases the rate and force of myocardial contraction, relaxation of smooth muscles of the bronchi. Epinephrine, or another name is adrenaline, is released from the adrenal medulla in response to flight, fight, fright, and exercise and hypoglycemia. Dopamine and norepinephrine serve as a neurotransmitter in brain and autonomic nervous system. Pheochromocytoma is a tumor which usually grows in the adrenal gland, which lie above the kidneys. It's also known as adrenal paraganglioma or chromaffin cell tumors. It is associated with VMA excretion, increased VMA increase, vinyl mandelic acid excretion, and epinephrine excess. Then neuroblastoma is another clinical condition. It is the type of cancer that forms in neuroblast. They are immature nerve tissues in the adrenal glands, neck, chest, spinal cord. Neuroblastoma often begins in the nerve tissues of the adrenal gland. Norepinephrine excess is most indicated feature in this neuroblastoma. Next clinical condition is Parkinsonism, which is associated with decreased level of dopamine in brain due to the loss of neurons in basal ganglia, and the clinical features associated with muscle rigidity, mask-like face, pinroll tremors, involuntary movements, and that is also known as Parkinsonism gait. So this is the tyrosine synthesis of dopamine. How it is being synthesized from tyrosine to dopa, that is dihydroxyphenylalanine. We saw in the previous slide also, and then from dopa to dopamine by decarboxylation reaction. So any of the step can be hampered. So dopamine is decreased or due to the loss of neurons in the basal ganglia, and the treatment is concerned with the intake of L-DOPA. Dopamine is a reduced form of L-DOPA. So L-DOPA intake will increase the concentration of dopamine. Then carbidopa and the L-methyl dopa is another choice of treatment, which inhibits the dopa decarboxylase and prevents the production of epinephrine. Methyl dopa is an antihypertensive. So biosynthesis of melanin happens in the melanoblast. It starts from tyrosine. Tyrosine, under the influence of enzyme tyrosinase and presence of oxygen, converts up into the dihydroxyphenylalanine. The same product is being formed, which is important for the synthesis of catecholamines. But here, there, the enzyme was a tyrosine hydroxylase. That was the basic difference, and that required the presence of tetrahydrobiopterin, whereas there is no role of tetrahydrobiopterin here and NADPH. So it is simply the one step, one enzyme dependent reaction, tyrosine to dopa, and the same enzyme converts dopa or dihydroxyphenylalanine to dopa quinone, and then dopa quinone converts up into the indole quinones, which further polymerizes into melanin. So just three-step process which lead to the formation of melanin, which is important for darkening of color of skin and hairs. So, so disorders associated with the deficiency of synthesis of melanin is albinism. The albinism is an inborn error of tyrosine catabolism pathway, and it is due to the inherited deficiency of enzyme tyrosinase, the enzyme which I just showed you. It is responsible for formation of melanin in the melanoblast. So this enzyme is lacking, and melanin protects the body from harmful radiations of sunlight. And copper deficiency also hampers the action of this enzyme, tyrosinase. So there may be disturbances in the pigmentation during the copper deficiency. Hair synthesized at the time of deficiency may be depigmented. So if copper deficiency is intermittent, alternate black and white regions may be seen in the hair, and that condition is flag type of hairs. Then clinical features associated are impaired synthesis of melanin results in either a hypopigmentation or no pigmentation of the hair, skin, or eyes that lead to the white hair and skin. Albinos are highly sensitive to sunlight and can lead to skin cancers. The lack of melanin pigment causes the fundus iris. So iris becomes hypopigmented. Photophobia, it is associated with nystagmus as well. That is a rapid involuntary oscillations of eyeball. Then skin becomes sensitive to UV light. So on exposure to light, there is an injury to the skin. Then melanoma, that is the melogen may be excreted through the urine in such condition. Then leukoderma, when the tyrosinase or melanin forming cells or both are absent from the epidermis. So leukoderma results. There is a graying of hair. It's also due to the disappearance of melanocyte from the hair roots. So there is a difference between albinism and leukoderma and vitiligo. Albinism, like depigmentation all through the body due to the complete absence or deficiency of tyrosinase enzyme. So all through the body, the melanocytes not able to synthesize the melanin, while in the leukoderma, in albinism, the all through the body there is a depigmentation. So in leukoderma and vitiligo, there is a difference. Vitiligo is self-generated. Here also there is a deficiency of melanocytes, maybe a deficiency of melanocytes or maybe the deficiency of tyrosinase, and it is self-generated and disorder. While the leukoderma may be accidental, maybe exposure to some burn, injury, or to some chemicals. Now let's see the formation of thyroid hormones, which takes place in the thyroid gland itself. So it is associated with four steps. First is the activation of iodine via H2O2, then attachment with tyrosine, then iodination of tyrosine ring, and then proteolysis of thyroglobulin. Thyroglobulin is a protein which is present in the thyroid gland cells, and the tyrosine residues of the protein thyroglobulin, they get iodinated via the active iodine, and then you can see it here. This is the monoiodine tyrosine is being formed, and here the two iodines are attached at the meta position. So this is the dihydrotyrosin. So two entities are being formed. Both conditions can be there, and then there will be condensation of these two entities. So if one monoiodotyrosine and diiodotyrosine condense, so they lead to the triiodothyronine, that is the T3. And if both the tyrosine units condense, they lead to the tetraiodothyronine, thionine, and that is also known as thyroxine, because here you can see there are four iodines, because these there is a duplication of these diiodotyrosine, matlab attachment of these or condensation of two such units, and here in the formation of triiodothyronine, one monoiodotyrosine unit and one diiodotyrosine unit are condensed, and they lead to the formation of triiodo. Means here in this hormone there are three iodines are. So that's how it is known as triiodothyronine. Further, after the condensation, there will be detachment from the thyroglobulin protein, which is all reaction is happening while attached, while tyrosine attached with the thyroglobulin. Now, once it is being formed, the hormones are completely formed. So there will be further detachment via proteolysis of the bond, and now these two separate hormones, they are in the system, in the follicles. So this is triiodothyronine and this is tetrahydrothyronine, that is also known as thyroxine.